Thank you. I'm actually Dr. Alperin. We have no disclosure. The current clinical paradigm holds that the childbirth-induced pelvic floor muscle injury leads to pelvic floor muscle dysfunction, which is one of the key risk factors for pelvic floor disorders. However, little is known about the mechanisms of pelvic floor muscle recovery after birth injury. We hypothesize that improper muscle regeneration is one of the reasons and producing mechanistic link between the muscle injury and the muscle dysfunction. We specifically aim to assess whether skeletal muscle stem cells are necessary for pelvic floor muscle regeneration after birth injury. In skeletal muscles, quiescent stem cells activate, proliferate, and differentiate in response to injury, fusing with the existing myofibers or forming de novo myofibers. This process is necessary for muscle regeneration. To assess the role of muscle stem cells in pelvic floor muscle recovery after birth injury, rats were subjected to local irradiation, which induced DNA damage and consequent muscle stem cell impairment. 1 day post-irradiation, the animals were subjected to simulated birth injury by vaginal distension, and pelvic floor muscles were harvested at 3, 7, or 28 days later. We first focused on assessing if the muscle stem cell quantity was affected by irradiation. In unperturbed muscles, the control group in irradiated animals did not differ in the number of muscle stem cells. This difference was also not present 3 days post-injury. In contrast, dramatic expansion of muscle stem cells occurred 7 days post-injury in the control groups. However, this expansion was absent in irradiated animals. We then went on to understand what are the mechanisms beside this quantitative difference between the groups. In order to do that, we assessed the expression of Ki67, which marks proliferating cells, by marking all cell cycle phases except G0. Again, in unperturbed muscles, there were no differences between the groups. However, at 3 days and 7 days post-injury, there were dramatic differences between irradiated and control animals, indicating that pelvic floor muscle stem cells cannot proliferate in irradiated animals. We then wanted to know what are the consequences of this functional impairment of muscle stem cells by assessing ongoing pelvic floor muscle regeneration through expression of embryonic myosin heavy chain, which is a marker of newly formed myofibers. In the control group, we see embryonic myosin heavy chain positive fibers in green appearing as early as 3 days post-injury. In contrast, there's a strikingly lower expression of embryonic myosin heavy chain in irradiated animals. Consistent with that, there was a significantly decreased size of the newly formed myofibers in irradiated animals compared to the controls. We then wanted to know what is the consequences of this difference by assessing ongoing pelvic floor muscle regeneration at a longer time point. This cross-section shows tightly packed myofibers of consistent size in a healthy pelvic floor muscles. Simulated birth injury induced the damage to the pelvic floor muscles evident at the earlier time points was progressively resolving by 28 days post-injury. However, in the irradiated animals with the functionally impaired muscle stem cell, abnormal pelvic floor muscle morphology was still evident 4 weeks post-injury. In summary, muscle stem cells are necessary for muscle regeneration. I'm sorry, this is frozen. When muscle stem cell function is impaired by either mechanism, in our case by inducing DNA damage, that leads to the absence of muscle stem cell expansion and proliferation, overall indicating the muscle stem cells are indispensable for pelvic floor muscle regeneration after birth injury. That's it. Thank you.
Thank you.
Could we do something with the computer again, please, just while we take the questions?
Okay, I have a question. Now, it's well known that a fistula in a previously irradiated woman will never heal. So is this a future treatment of these patients, you think?
No.
With the stem cells?
Oh.
Yeah.
Sorry, I meant the irradiation, which certainly would
No.
No. Of course, I mean, this was not for the vaginal connective tissue.
No,
this is
know.
just purely for this is muscle stem cells, known as satellite cells as well. I think one of it is, there is certainly, my lab is and people in my lab, we focus on understanding how we can potentiate endogenous regenerative potential of these muscles by potentially affecting stem cells. But it's also, I think, an important mechanistically because we know from epidemiologic data from Scandinavia and US and other countries that aging, for example, women who deliver at an older age, they recover worse. And so we don't really know why it is. Is it possible that they're closer to the development of pelvic floor disorders through other factors, like hormonal deprivation, et cetera? Or is it truly that their reparative process is not as good? So I think that it's kind of a two-pronged information by potential future therapeutics of harnessing the endogenous potential, but also to understanding what are the risk factors for the poor recovery after delivery.
Lovely. If there's no more questions, thank you very much.
Thank you.